Welcome to another edition of CCSD Living Environment. Today we're going to look at DNA replication. We're familiar with cell division, and part of the idea behind cell division is that we need to make sure that we have an equal amount of DNA in both cells. And to do that, the DNA needs to replicate. So we need to look at the process that the DNA uses to actually undergo replication. So DNA replication is the process where DNA is copied. We actually make a copy of the DNA molecule. The first thing that happens is an enzyme called DNA helicase unwinds and unzips the DNA molecule. So think about that double helix. We're going to unwind it, so we're going to put it back in that ladder organization, and then we're going to unzip it. And just like with a zipper where it pulls apart in the middle, so again, right, here's my zipper, just like a zipper, how it pulls apart in the middle, that's what we're going to do with the DNA molecule. It's actually going to come apart in the middle. Now remember, we said that there's weak hydrogen bonds in the middle. So those weak hydrogen bonds are going to open up, and what does that expose? That exposes the nitrogenous bases. So once we do that, each parent or original strand of DNA serves as a template to construct a new complementary strand. So if I have A on this side, what do I get on this side? T. G on this side, what do I get on this side? C. C on this side, G, and then T on this side, I get A. So it gives me the complement as it goes through to fill in. The new strand is assembled from free DNA nucleotides that happen to be floating around and hanging out in our nucleus. So the question comes, where do these DNA nucleotides come from? Well, think about it. When we eat food, the foods that we eat are living things. Those living things also have DNA. So as part of the process of digestion, as you're breaking all this stuff down, those DNA nucleotides get released. And they end up in the nucleus of the cell, and those nucleotides get reused to give us the new molecules. And it's just like with amino acids or glucose or anything else. Remember, DNA is a polymer. So we need to use those monomers, the nucleotides, to build the new DNA molecule. So what happens next? An enzyme called DNA polymerase III, DNA polymerase. I know it's an enzyme, it ends in ase, polymer. It's the enzyme that makes the DNA polymer. It'll match the bases on the parent or template strand one by one with the new bases. So A, I'm going to bring in a T, G, I'm going to bring in a C, C, I'm going to bring in a G, C, I'm going to bring in a G, and it does it for the whole strand. The free nucleotides are connected to the original nucleotide, again using those weak hydrogen bonds. That's how we reestablish those rungs down the middle. Strong phosphodiester bonds, those strong bonds, or sugar phosphate bonds, form between the nucleotides to create the new backbone or the uprights of my ladder. So first it's going to connect those weak hydrogen bonds, then I'm going to reconnect my strong bonds, those phosphodiester bonds. Finally, I end up with two identical strands of DNA. So I have one strand, two strands. Because remember, I had one strand that opened up, both of them got filled in, and that gives me two new strands of DNA. Worth noting is the fact that one DNA molecule replicates to produce two DNA molecules. And each replicated molecule has a strand of the parent DNA and a strand of the new complementary DNA. So what I've tried to do is represent that for you in a picture. Here's my original parent strand of DNA. When that gets unzipped, when that gets opened up on those weak hydrogen bonds, it's then going to match up to a new complementary piece. So if this was A, this would be T, C would be G, G would be C. So the other half of my original strand is here, and again, those complements are going to fill in. So my one strand will give me two strands. So what they refer to that as, DNA replication, is what they call semi conservative. Semi-conservative because half of the DNA molecule stays the same. The other half is all part of the new molecule that we build. But this strand of DNA is identical to this strand of DNA, which is identical to this strand of DNA. Remember, we don't want any changes in the DNA. We want every part of it to be exactly the same as what we started with. That takes us through DNA replication. Next time we'll start looking at what do we do with DNA? How does DNA actually turn us into what we are? Thanks for watching.